And do you see it so entertaining? Like, do you think that one day when you stuck all the answers?
No,
It was great.
I did. It was bad. One was back on himself. I did mine at the back. That's kind of what I figured. I read the same time. Yeah. I don't know. 90 seconds. I think you just kind of like the wrong temperature too long. So I told you to get it. I had to see if the change in temperature. Sorry, you guys Let's do 5 degrees off.
Did you just say that means California? I didn't.
So we ended last time talking about amphibian bromide. Okay, so I've been drawing this up here pretty much every day this section. Might be important. So we left talking about intercalating agents. Amphibian bromide is not the only intercalating agent out there. What intercalating agents do is they get into the DNA and then they induce frame shifts. I don't know how, but there are apparently some intercalating agents. Is that camera on? Okay. Some intercalating agents that can cause deletions, but generally they cause insertions of 1, 2, or 3 nucleotides, depending on the size of the molecule as it intercalates into the DNA. Do you guys have any questions about intercalating DNA molecules? This is why I kept telling you don't eat the amphibian bromide in lab, right? Okay. So, let's talk about mutations. Let's talk about a point mutation. When I say point mutation, what's another word for point mutation? Substitution. Some people incorrectly use it to say point mutation, deletion, point mutation, insertion. It should mean substitution. So let's talk about a substitution. And there are a couple different forms of point mutations, substitutions. We were talking about the redundancy in the code last time and how we could, or a couple times ago, we were talking how you could use that in microRNA treatments, right? You could knock out the normal gene, you could use the redundancy to insert an alternate sequence of DNA that would encode for the same amino acid, right? So silent mutation is a change in the DNA sequence, but the amino acid code isn't different. Does that make sense? So, I'm going to propose this to you. I'm hoping some of you guys get your thinking caps on today. How could I get a point mutation, which is a silent mutation? It's not going to change the amino acid sequence of the protein. How could that affect the protein? Maybe the protein is not expressed at all. How could it possibly be that a point mutation could affect, a point mutation which is a silent mutation, could affect whether the gene is expressed or not?
Maybe it has something to do with it gets in the way of some kind of RNA processing.
Okay.
Maybe it's just a really important spot.
Gets in the way of RNA folding or processing.
Yeah.
What's the types of processing we talked about? Splicing. So if we had a substitution, we think back to our splicing. We've got exon, and we're going to and we're going to cut at the G, T, R, A, G, T, right? Then this is all the stuff that's cut out, C, A, G, G. So this G is going to wind up getting connected to this G over here, right? And all this stuff gets thrown away. So this is part of the code, right? So if this is one of the nucleotides in the amino acid, actually that wouldn't be it, we'd have to go like this. If that's the codon that's going to be in the chain, so methionine, something, 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 what's AGG, I have no idea, we'll just call it alanine. It's not alanine, but we'll just call it alanine. If I get a point mutation, substitution, that changes this, say, to a C, now this is not a very good splice site. the C could still code for alanine in this codon, right? So you would look at the genome and say, oh, it doesn't affect the amino acids. It shouldn't be a harmful mutation. But splicing could be screwed up so that this site is not recognized as the other site. Maybe it recognizes up here, and you start translating from here down, and you get the wrong amino acids. Does that make sense? Good. Okay. That was easier than I thought. You guys all figured that one out. Alex is just the one who verbalized it out loud, right?
Yeah.
Okay, great. So, a missense mutation. Oh, here it is. I got the code right here. ACG, AGC, AGC, or serine. My bad. Anyway. A missense mutation is when you change one amino acid and you change one nucleotide, you substitute one nucleotide, and you get a different amino acid in it. Right? And a nonsense is when you convert an amino acid that codes for something into a termination codon. So if we had a sequence that we came along, and suddenly in the sequence, maybe a GGA, which codes for GGA glycine. So if we get a substitution, which is a point mutation, and that G is converted into a T instead, I think this is on its last slice, TGA now codes for a stop codon. So all of the final amino acids on the downstream portion are not going to get translated into that protein, right? So it's a nonsense. You come to the end of the protein. Premature termination codon. Okay, those are different types of mutations we have that are substitutions. You know when an insertion and deletion, if it's a change in nucleotides of 3, it stays in frame, right? What happens if you insert 9 nucleotides? You'll add 3 amino acids. And in general, I'd say probably the protein's going to be okay. Depends where it happens. If it happens right in the active site of the protein, probably screw up the active site of the protein and ruin it. But most of the protein is not active site, so the protein might be able to absorb those 3 extra amino acids and be okay. Okay. So that's damage to DNA. Usually that type of damage is repaired incorrectly. Now we're going to talk about bigger types of mutations. And these are chromosomal alterations and then alterations in the number of chromosomes. Right? So chromosomal alterations. Here's a classic example of this. When does crossing over happen? Are you not raising any stretching?
Well, prophase 1.
Prophase 1, yes, absolutely. Everyone was thinking that, right? In meiosis, crossing over happens, right? And what we talked about is we have a chromosome like this. So I didn't draw it well. And then so if this is from mom, the chromosome from dad lines up. And the sequence lines up. Right? So sequences on here actually line up and then physically cross over. Such that, and when I talked about it earlier in the semester, I probably said, for the purposes of this class right now, the chromosomes line up exactly right. You've got a zipper and zipping up properly. I want you to imagine if you had a zipper and normally you would zip up from here. What if you started the zipper, you aligned them incorrectly? Right? Imagine if you could take part of the zipper and slip it down through, and you could start, I should have brought my jacket, and you could zip it, so this one was halfway up and this one was halfway down. Can you imagine that? Who here is OCD and is cringy as I talk about it? Yeah? how could, based on what we were talking about before, how could a chromosome align incorrectly? Like that. How could a chromosome align incorrectly?
Some kind of mutation. Make it look like something where it's not supposed to be.
And does it require a mutation?
Could be.
What could cause two chromosomes to line up incorrectly in prophase 1? Which I guess isn't incorrectly.
Duplicated stretches of DNA.
Duplicated stretches of DNA. How
Like
does that
the
happen?
ALU sequence.
A repetitive DNA sequence. So what happens is the nucleotide sequence on one part of the chromosome lines up with the nucleotide sequence on another part of the chromosome. So if this sequence is ALU, and it appears 1,000,000 times in the human genome, we've probably got ALU in the proper spot on the other chromosome. We might have ALU on another spot on the same chromosome. So that 350 nucleotide sequence can line up and we go, oh look, my sequence matches yours. Great, let's cross over here. Oops. That doesn't work out so well, does it? If homologous chromosomes line up incorrectly like that, when crossing over finishes, so here they're aligning incorrectly. Apparently A has the same sequence as C. In crossing over occurs, one chromosome winds up reading, instead of A, B, C, D, E, F, G, it reads A, B, C, B, C, D, E, F, G. We've duplicated B and C in this chromosome. And this one is screwed, it doesn't have B and C. So then we wind up with a chromosome that is, it's got extra nucleotides in here, and this one is missing. This is an insertion, and this is a deletion. What is the word for, what's the term we use when someone drops a nuclei onto a glass slide and the chromosomes go splatting out and you get this field of them? Karyotype. Karyotype. So you can purify the chromosomes that way. You can stain them with this DNA stain called GIMSA. And that's how you get the banding patterns of the DNA. So that in one chromosome, you get this is the same as this. And the other chromosome would be missing that. Right? This compares to this, and this guy is apparently missing from this spot right here. Right? So early on, before we could sequence a human genome like that, we still can't do it like that, but it takes an afternoon. That takes forever. This is how we would detect chromosomal abnormalities. Is people would do these GIMSA stains. Now people can do sequencing. Yeah.
How often does this happen in a cell?
How often does this happen? I don't know a number to that.
Because if this happened in a cell, wouldn't that cell just die? Or
No.
that's what disorders, some disorders.
Right. So if this happened in a cell, that's a great question. If this happened in one of my skin cells, that cell's just going to die, right? No. Why not?
Maybe it doesn't code for central stuff.
I mean, this is a huge part of my chromosome. Let's say there's 100 genes on here.
Because one of them won't be expressing it, but the one that was duplicated will overly express it.
Yeah, I have the same number. I have the same copy number of everything.
Yeah.
I've got the exact same amount of DNA in here, so it works. Does that make sense? Now, we're going to see in a minute an example where it could get screwed up and where it might die. But that's kind of a finesse little detail. But in general, if this happened, say a fertilized egg had a chromosome, a rearrangement that happened like this, such that every cell in the person's body was like this, person's probably fine. Now, what happens when that person goes to reproduce?
Their offspring might not be fine.
Their offspring may not be fine. So, let's say this is me. Let's say we don't know, but this is chromosome 3 on me. This is huge. Chromosome's 3 big. And I've got duplications on this. I could be fine. But the sperm that I would be producing would have either this chromosome or this chromosome. So that's the fertilized egg is missing either 100 genes or it has 2 copies of 100 genes. And that's where problems occur. So you guys haven't yet reached this point in time in your life. But at my age, I'm 44. Everybody, I went to college twice. Everybody I went to college with is all done having their kids. And then I went to college a second time to get biology, because psychology wasn't working for me. And all the friends I made there are now reaching 40 and done with having kids. And so I had you guys had your close group of friends. You go and you have a party, or you go hang out, or they're in your fraternity or sorority or something. The people you go hang out with all the time. In each of those groups that I've had that twice independently. I've got 2 distinct groups up there. They're my close friends like that. There are individuals that have, it's been murder, but they've tried and tried and tried to have kids. You know, it's hard work. And it sounds funny now, but it probably sounds funny to you, but they tried for decades to have kids. Never happened. And in some cases, we don't know why. But for me, my guess is some of my friends may be like this. They may have this type of chromosomal rearrangement such that the egg that gets produced, if that egg gets fertilized, it doesn't have a full complement, or it has too much of a complement of genes. And the genes getting expressed cause it to develop incorrectly, and it aborts. It spontaneously aborts. It's miscarriage. So you guys don't understand this yet, but people who, when they're trying to have kids, every month that goes by and the pregnancy doesn't happen, it's devastating to these people. I mean, imagine that for a decade, working to have a kid for a decade, and every month it's just totally blows people away that it's not happening again this month. So it's pretty sad, I have one college friend, she wanted to have kids really, really badly. It just never happened, so she's kind of living vicariously. She takes her nieces and nephews as often as she can. That's how she's experiencing the joy of children. So that's a long way of getting is, I think it's probably more common than people are aware.
Is it expensive to slot your DNA on a thing?
Yeah, is it expensive to get a karyotype made?
Yes.
So when people used to do karyotypes a lot, it was a couple $1,000, several $1,000s, and I don't think insurance would say, you go to your insurance company and say, we're not able to have kids, and they go, bummer. Why would we pay a couple $1,000s to figure out if you're ever going to have kids or not? Keep trying. If it works, it works. If it doesn't, it doesn't. Now, doing karyotypes, people would just do the sequencing. There's sequencing ways to do it. Probably you could get it done for $1,000 would be my guess. It'd be fun. Right place. I don't know the details on it. It shouldn't be very hard. In theory, a student here could do an independent study, figure out how to do that test by PCR, could isolate their own genomic DNA and run the PCR in the lab up there and get those results and determine it. Okay. Any other questions on this? And that's exactly it, the ALO sequences. The cause of a lot of this. What if chromosome 1 and chromosome 7, if ALO sequence had chromosome 1 and ALO sequence had chromosome 7 aligned up, and you crossed over? So earlier we were talking about chromosome 3 and chromosome 3 getting this recombination incorrectly. What happens if it's between 1 and 7? Is that cell going to be viable? You're shaking your head. You think it is? Yeah, it will be. Because again, they have all of chromosome 1 here and all of chromosome 7 here, right? They still have a full set of DNA. That person or that cell is fine. If a cell with this is used to produce sperm or egg, now that egg is going to be missing some DNA. These are not going to segregate properly. And the egg or the sperm will probably be missing or have an extra copy of the chromosome. I'm going to turn the lights off real quick. Sorry about this. This is a karyotype in which they actually show this has happened. They've colored one chromosome in red, and the other chromosome, here's a chromosome in green, and here's a chromosome in red, and here's a chromosome with green and red, and here's the green with a little red on it down here. That's an actual karyotype. Okay, lights back on. I hope that didn't screw up the lighting on the camera real quick. These things definitely happen. In addition to homologous recombination going wrong, we've talked about X-rays can induce these double-stranded breaks. In DNA, in general, when DNA is hanging out there and doesn't have a telomere on either side to protect it, the DNA kind of freaks out and goes and finds any other piece of DNA it can connect to it. So if someone's been blasted with X-ray radiation, their chromosomes may have all been broken into bits, and the DNA may shuffle all around and reconnect in odd ways. So here are some terms that they describe. We talk about duplications, so insertions and deletions, duplications. We've talked about these conceptually before. But there's some other changes. There's a translocation, if you get non-homologous chromosomes recombining. And then there's an inversion, where and this usually would happen in a chromosomal break in like an X-ray situation. You break here and you break here. This thing goes floating around, and before it connects back in where it came from, it actually spins around. So instead of reading 1, 2, 3, 4, 5, 6, 7, 8, it reads 1, 4, 3, 2, 5, 6, 7, 8. That piece of DNA physically spun around that way and then connected it. That happens. And usually, totally fine. The person's totally fine. And in this case, the chromosome probably segregates properly, and any fertilized egg with one of these in it would probably grow normally. The person would be totally fine. Okay. Let's get into the finesse details to your question, would the person be all right? Or how often or something wasn't you, or someone else asked, would the person be all right? So let's take a look at one of these locations. Let me go back here. So let's do this duplication. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 2, 3, 4, 5, 6. Actually, let's do this one here. Non-homologous recombination. That's easier to see. So we wind up with one chromosome that's got part on some other chromosome like this. Let's look at this junction point right here. In this junction point, we've basically got this chromosome here with blue. I've notated with blue. Here's the +1 this is the promoter. This is transcription start. So this is going to be the pre-RNA that's made. And this is the coding part of the RNA, right? That's the amino acid coding part. Everyone understands that, right? You've learned the system of understanding these figures, yes? And then this is say chromosome 1, and this is chromosome 7. Here's the promoter for some other gene, +1. And this gene is expressed very strongly as a big arrow. This gene is not expressed very much. It's a really small arrow. That's another notation on this. The size of the arrow tells you how much the gene is expressed. So let's go back and what would the difference be? Remember this? Glucose, pyruvate, right? Call that glycolysis, right? All fun. What's the first step? What's the first enzyme that does this? Hexokinase, is that right? So every cell has to break down glucose like this. So every cell expresses a whole bunch of hexokinase. It's a big arrow for hexokinase. But for a cell to go through cell division, it only needs a little bit of this protein. Let's get a non-homologous recombination. You can say there's an allele sequence somewhere here and somewhere here, such that we recombine non-homologously. That we wind up with a promoter from the biochemical hexokinase. And we even have a little bit of amino acid for hexokinase. But that happens to be fused, the recombination occurred here, such that most of this gene, this exon didn't make it. But these exons got transferred over. So what will happen is, RNA polymerase will say, hey, here's a promoter. It'll reach out, measure the proper distance out, and it'll say, oh, it's a really strong promoter, so I make a whole bunch of RNA off of this. So it makes this pre-RNA. This pre-RNA gets spliced. Oh, this thing doesn't explode on me here. So this is an exon, and then it's going to get spliced to this exon, right, with the blue on it here. And then that gets spliced to this exon, which is red. Let's do it this way. We'll call this red. Right? So what we're going to wind up with is this. Once the splicing is done, here's my methionine. So the protein is going to be this. It's going to start with the methionine here. It's going to read down, and then it's going to read right into this until we hit our termination codon. What are the odds that this is in frame with this? 1 in 3. So there's a 1 in 3 chance that this protein is a chimera or a fusion protein. What's a chimera? Does anyone know? Any of you Dungeons and Dragons people out there know what a chimera is? I'm embarrassing you now. Part what?
It's like lions, ancient eagle.
It's an animal from ancient Greece. That's part lion, part eagle.
Snake.
Part snake.
There's a lot of different uses. Some are less specific.
So a mermaid is sort of a chimera. It's half fish, half human, right? So this is a chimeric protein or a hybrid protein. It has a little bit of amino acid for hexokinase, and the rest of the amino acid is for this thing that says, cell, go ahead and divide. Divide, 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 divide. Now I did this in a very specific manner when I drew this out, is I have so much, I have almost the entire amino acid sequence of the cell division gene that this protein probably folds up properly and works. What's the problem?
Getting way too much of it.
I'm expressing way too much of it. So the cell is getting this real strong signal to divide, 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 and the reason the signal is so strong is RNA polymerase says, oh, let's make some hexokinase. And it makes this divide, divide, divide gene instead of hexokinase to process glucose. this is where you get problems. This is where you get a skin cell, which had this happen, and the skin cell is not normal. Now you get a skin cell that divides and divides and divides and divides, and we call that cancer. It's a tumor.
How do you get enough nutrients and everything to build up the cytoplasm and mitochondria and everything that needs to divide every time and form a viable cell?
Great question. So we'll get to that. That's the last bit of this course is talking about all that. I'll give you a summary of this. If you have a cell that's being driven to divide and divide and divide, such that that cell doesn't grow large enough that the progeny can survive, that tumor cell, by natural selection, dies off, and you don't get cancer. It is only a cancer cell that gets stuck dividing and dividing and dividing, and also finds a way to bring more nutrients in, that is able to persist to become a tumor. And it's natural selection. If this happens and you get a cancer cell, I call it a pre-cancer cell, dividing, and it doesn't pick up the other ability, natural selection says you're gone, you're out of here, you don't survive. Does anyone know what does angiogenesis mean? Does anyone know what that is? You know what that word means? Generating new blood vessels. So a successful tumor, once it grows to a certain size, it needs so much energy, it won't be able to grow anymore unless it also turns on an ability to somehow trick blood vessels to grow into it and give it the nutrient supply that it needs. It's unbelievably how hard it is. The odds of cancer developing in you is so small, it's unreal. It's just crazy. But you have 10 trillion cells. When you get to a large number, and you've got 10 trillion cells lasting 44 years, that's a long time for all of these screw-ups to all happen in just the right cell to get cancer. It's just a question of odds. Good. So, these types of rearrangements, when we take a cancer cell and we do a karyotype on a cancer cell via the genome, finding these non-homologous recombination events is common. They routinely happen. I'll give you an example. Somebody, very, very early on, using the karyotypes, said, they didn't even know that this could happen. They said, I wonder if this could happen. If this happens, I would expect that where that recombination event happened, if I could find out what gene that recombination event happened in, I could identify the genes that are causing the cancer in the person. And so this person went out and got a B cell tumor from someone. It's a lymphoma. They took the B cell and they basically did this chromosome spread and they found a translocation and sequenced that stuff in there. And this is the first B cell lymphoma they did it to. That didn't work out. And so the gene that they identified, they called BCL1, B cell lymphoma 1, was not anything involved in cancer biology. It was just a recombination that happened. They got a different B cell lymphoma from a different person and said the site of recombination, whatever that gene is, we're going to call that BCL2. And when they characterize BCL2, BCL2 is it. If you want to become a cancer cell, you had better find a way to activate BCL2, that gene. You have to increase the expression of it if the cancer cell is going to survive. We'll talk about why later. So if I can actually make my work. Yes. All right, one down, so like 30 more to go. I'll get all of your genes, everybody, your gene will be talked about, except for the TMEM ones, because we don't know what the hell they do. Yeah. I told you, they localize to the mitochondria. We don't know what they do there. Okay, let's talk about alterations in chromosome number. These are changes where one chromosome, the structure of a single chromosome or two chromosomes is different. Now what we're talking about is instead of a creature being 2N, it's 2N plus an extra chromosome. And that's aneuploidy. Or 2N minus a chromosome, which is kind of rare, but it happens. Sometimes a female could be missing the X chromosome. I think it's called Turner syndrome. So, aneuploidy is loss or gain of a single chromosome. Generally, that's pretty bad for the cell, and the cell, quite often these cells aren't going to make it, they won't survive. Depends on which chromosome it is. And then the term euploidy is gain of a whole set of chromosomes. So, classic example of aneuploidy is trisomy 21. There can be a person that inherits an extra copy of chromosome 21, or very early on in cell division winds up with a cell that has an extra copy of chromosome 21. Chromosome 21 is a very, very small chromosome with not a heck of a lot of genes on it compared to some of the others, such that cells can survive and the person can grow and develop, I'm going to say normally. You guys decide what's normal. Is this normal or not? And I think these people generally, some of the Down syndrome can survive maybe until they're 40-ish or so. The severity of it differs. Some people are pretty well incapacitated. They can't go out and work and provide for themselves. Some people are very, very high functioning. Go out and work and have a job and live in an apartment on their own and be just fine. So when you do a karyotype of this person, that there's actually 3 copies of chromosome 21. There was a case, someone with Down syndrome, so they just did the classic test, they did the karyotype, and they found 2 copies of chromosome 21. How could this person have Down syndrome with just 2 copies of chromosome 21?
So like recombination where part of chromosome 21 was on a different chromosome?
Recombination where part of chromosome 21, so they had 2 copies of chromosome 1 and then they had some other chromosome and a piece of chromosome 21 was on it. Or maybe all of chromosome 21 was on it. That's great. That's not what happened in this case, but yes, that's very possible. That could be an explanation.
Maybe the cell that they looked at was a mutant, and for some reason during...
Sure, maybe the cell, so that's often the case, is sometimes fertilized egg has 3 chromosome 21s. And during the cell divisions, some of those cells get back to a normal number of 2N, such that part of the organism grows, part of the person grows trisomy 21, and other cells in that person grow 2N, normal human, what we would think of, right? That happens definitely. There are instances where the person looks trisomy 21 physically. They have the appearance, the stubby kind of appearance of trisomy 21, but their brain is 2N. I want you to imagine going through life, everyone saying, "Hey, how are you today?" And you're normal in the head. You just look trisomy 21.
Can the opposite happen, where you look normal?
Yeah, you can look normal but be trisomy 21 in your neural tissue.
Is it normally just appearance and neural tissue?
No, there are other things. Yeah, there are some other things. I remember liver or kidneys. There are some other things that happen. So that's very possible. What's another possibility?
I think it's called mosaic Down syndrome, and you get the normal amount of chromosomes from both of your parents, but then as your cells divide, that's when the mutation occurs, so it can occur really late.
So what happens is the fertilized egg has 2N, and it's dividing and dividing and dividing, and one of those cells divides and gets 3 21s, and one cell only gets 1 21, and this one dies off. This is missing genetic information. But this one survives, and again you get a mosaic situation. In that case, most of the organism would probably be your typical 2N, and then some of the organism would grow, some of that human would be trisomy 21. Probably there are parts of your body that are aneuploid like this. Everyone probably has it to some extent. Yeah.
Do you know if it would be possible to tell when in the cell divisions that had occurred?
Is it possible to tell when in cell divisions it occurred? So the one thing, so the sequence of chromosome 21 you get from your mom is a little bit different than the sequence you get from dad. That's natural variation. So we can determine which chromosome, from mom or dad, which chromosome is the extra. I think based on DNA methylation patterns, they can tell if the chromosome you got from your mom, if the trisomy 21 was inherited or developed in you. I think there are ways to determine that. But as far as, I mean, there's patterns. We know to a certain level when cells divide, we know like when you're at the 5,000 cell stage, you know that that cell is going to become the liver. And if your liver is trisomy 21 and the rest of you isn't, then you know they can make that guess. Okay, let me tell you about this particular case. Person with Down syndrome. Had a duplication on a small part of chromosome 21. When you looked on a karyotype, that's what you saw. You guys see the difference? There's an insertion right here. That's the duplication. When you looked on a karyotype, when they looked at the karyotype, the chromosomes looked the same. The banding pattern was the same. They looked the same size. The insertion was so small, it contained 5 genes. Trisomy 21, Down syndrome, is caused when you get a duplication of 5 genes. You don't have to duplicate the whole gene, the whole chromosome. Duplicating just those 5 genes is enough. How about that? Dumb luck? Okay. This is my favorite. Oh man, I love this. When we get polyploidy, euploidy, an organism, this would be if in the process of me making a sperm, all of the chromosomes went into one sperm, such that one sperm was 2N, it's supposed to be 1N, right? And the other sperm was 0N. It didn't get any chromosomes. All the chromosomes went over here. And that 2N sperm finds an egg, and in my wife, the same bizarre thing happened to make a 2N egg instead of a 1N egg. If my 2N sperm fertilized her 2N egg, there's a matching chromosome for everything, and you wind up with a 4N organism that grows and develops. In humans, to the best of my knowledge, not possible. It would die out. It would die. It wouldn't develop properly. In plants, this polyploidy happens, and the things are fine. The classic example of this is wild strawberries, things that are the natural strawberry things that are out there. Are these dinky, this is real size for a strawberry. I may have told you guys, did I tell you guys a story already about strawberries?
Not in this class.
Not in this class. So a really good friend of mine was like, oh, I know where there's this patch of wild strawberries out along on this trail. We'll just go out to the area and we peel off and there's strawberries. And it's about the right time of season, you know, it was like around July 1st or something in Michigan, northern Michigan. So yeah, that's perfect strawberry time. So I was like, awesome, let's go out there and we'll just sit there, we'll be up on this cliff looking out over the water, it'll be awesome. We get out there. And the strawberries are these, this is how big a wild strawberry is, that big. You spend 15 minutes picking strawberries and you go, that was a great bite to eat. I was pretty disappointed. People who were growing strawberries found a strawberry one time that was enormous. These, when I was a kid, this is the size of a strawberry. You go to the grocery store. These are strawberries. You guys look at me like, what the hell kind of world were you living in? Right? So a polyploid event happened. These guys are 2N. These guys are 4N. In my lifetime, someone was in their field and they found a strawberry that was polyploidy happened again, where this thing is 8N. You know what I'm looking for in my lifetime?
Oh my God.
In your lifetime, you will not be buying strawberries in a container. You will buy strawberries like you buy apples. In your lifetime.
Like watermelons?
Or watermelons.
This thing's huge.
256N. Let me turn the lights out. I really think we all need to take a look at this one.
That looks like an amazing
Is
strawberry.
that
That is an amazing strawberry.
But how big are the hands?
I don't know. They look like small hands to me.
Exactly. You can lie in pictures.
Sure.
Still, that's
Like
a huge
5-year-old
strawberry.
kid.
Does it affect strawberry flavor at what size they are at?
I don't know if it affects the flavor or not. The strawberries that I've been eating this week, I don't know. It's strawberry season somewhere. And the strawberries that we ate this week, my wife went and got a container of them, and we ate them all. The family just sat around and ate the whole thing right there at the spot. She's like, I guess I'm going back to buy more strawberries. And we now have a shelf, of the bottom shelf is filled with strawberries in my house. So it is strawberry season somewhere. It's really tasty.
Are they on sale?
It doesn't matter if they're on sale.
When you're poor, it does.
So yeah, fun things happen like this. Good stuff. Okay. Wow. I didn't get very far today. We'll get into DNA repair next time. Lab. I've got 2 minutes left. On Thursday, what we decided to do in Thursday lab is, it's the end of the semester, we're done. Don't set up additional reactions. If you have something that you're in the middle of, finish that up. Otherwise, your job now is to go back to your notes from the computer portion of lab, get all of those notes in an order where someone who has a whole crap load of grading to do and doesn't want to do it can just page through and see all of your work. Oh, it's all underlined, and there's a note over here that says underlined is the coding portion of the sequence. And the italics means it's the initiation codon or the termination codon. Nice and easily annotated, so that when I get to the end I go, that was really a lot of fun, I think that's an A. Because if I need to dig through all this crap, I'm like, I don't understand this, it doesn't make sense, what's on this page, there's nothing labeled, someone threw this together. Wednesday night at 4 in the morning, and here I am grading it, grouchy that I've just gone through 20 of them and I'm not in a good mood. What kind of a grade do you think that gets?
An A minus.
An A minus. So organize all that. And I want you to organize it in a manner you think is sensible. Present it to someone. Remember when you walked in there and you were doing all this and you're like, I have no clue what Bog's asking me to do all this crap. I'm glad he's got these instructions and I'm kind of following through this. Do it to someone who's going to come in next year and take your spot, basically. Have all that information that way. And then for the wet lab portion, you should have a picture of your gel, and all of you, your gel failed the first time. Here's my gel, shows primers, and I failed. And I have no idea why, because I didn't have controls. And you should write all this stuff out in your lab notebook. So what I need to do next time is run controls. The controls I decided to run were this, this, this. When I turn the page, you should see the gel with those controls. It says, here's this and this and this, and this is how it came up, and this is what it means. My experiment still didn't work. What the hell? But at least I know that it's the tack that's bad. So I need to do, the next time I do this, I should have fresh tack. I turn the page, and it says, here's my reaction where I run fresh tack. Right? Here are my controls to test all this out. Nice and easy, so that I just page through it, 30 seconds I've graded all your lab work and you got the A for 20% of the class. Wouldn't that be great?
Yes.
Yes. Okay, so that's what I'm looking for. I will be in lab if you have things you want to be working on. I will open up the door right now. Yes? What do they do? I don't know. What do they do?
Finals week.
Finals week.
Thursday of Dead Week.
Thursday of Dead Week?
Finals week.
Thursday of Dead Week? That's a week from this Thursday.
Yeah. Not allowed to do this on
A week from this Thursday. No? You can turn it in early.
Are you sure if that like Tega reaction?
I'm giving you 10 days, 11 days. If you can't get it in, then let me know. Let's plan on a week from Thursday if you can't get it in, let me know. Yeah.